Thank you very much for having me, and thank you everyone for being here. So today, as the title of my talk suggests, I'll be talking about vaginas, okay? And so at Virginia Tech, I lead the STRETCH lab. STRETCH stands for Soft Tissue Research, Experiments, Theory, and Computation by Hawkes. So I'll tell you a little bit. My hope is to give you a very quick overview of the things that we do. Alrighty. So the first thing I want to clarify is that when I started to work in this field a few years ago, I realized I got very worried. I got very worried by the fact that there was really nobody working into the mechanical property of the vagina, despite the fact that this organ is very important, and despite the fact that mechanically it does really a fascinating job at actually stretching and deforming significantly over very short time intervals. And the other thing I want to clarify is what the vagina is, because believe it or not, especially when I talk with engineers think that, well, not only engineers, but people who are not necessarily familiar with the anatomy, they think that the vagina is the external genitalia. So they usually think that's the vagina, but actually the vagina is that tube that connects the external genitalia to the uterus. And if you look at the structure of the tube, this is a cross-sectional view. It's a highly inhomogeneous material with different layers. So it's a very, very complex structure. Primarily, I want to emphasize here, two layers. This layer here, what's called the lamina layer, and the adventitia, which are primarily giving the structural support to the organ. And then these two inner layers that are basically made of smooth muscle cells that actually cause the contraction of the organ. All right, okay, so why do we need to worry about vaginas? Well, the first thing I want to show a few stats. The first one is the maternal mortality rate in US, which is very high, and you can see here it's not going down, and you can also see how it compares with other countries. So we're doing very poorly. It seems like there are more women dying because of pregnancy in US than in other developed countries. And unfortunately, the number is not necessarily going down, okay? Then the other thing I want to talk about is how women who actually have babies are more prone to have pelvic floor disorders. And these include basically urinary incontinence, fecal incontinence, prolapse when the organ basically fall out of place. And this figure here shows the difference between not having baby, having baby vaginally, and having baby through a C-section. And it looks like having a baby through a C-section can prevent some of the trauma that we undergo during a vaginal delivery, but the C-section is still a surgery and has complications. And unfortunately, even when you look at C-sections, what you see is that in US, the rate of C-section is going up. And there are specifically some states where the rate of C-section is higher. And that has nothing to do with women needing more C-sections. It oftentimes has to do with where you have your baby, which hospital you're going to have it, and you're going to see that the rate of having a C-section is higher. So we still don't know when a woman should undergo a vaginal delivery versus a C-section. There is no scientific-based evidence that suggests that some women have to have a C-section. All right, so the question that I wanted to answer in my lab was like, how much does this organ deform? Okay, do we know? And unfortunately, oops, sorry, something happened to my slides. My apologies. I clicked on the page. Unfortunately, we still don't know how much this organ deforms, and clearly it deforms significantly during copulation, but there are deformations that occurs just because you're running, you're walking, everyday type of events, right? And so in our lab, what we do to measure this deformation, we use animal models, and so we use a lot of rodents, right? And you can see here basically the entire reproductive system, and this is the vaginal canal. They're very, very tiny little tubes. Look at the dimensions right here. So 12 millimeter in height and 2.6 inner radius. So very, very tiny tubes. And what we do is we develop our own setup to test these vaginas. And basically what we do, oh no, I hope this is not going to happen. So what we do is basically we clamp the vagina between two needles, have a pressure pump, inflate water, and then we speckle the vagina so that you have basically speckles that you can follow as the pressure increases within the organ. And so this is a way of measuring the actual deformations of the organ. Here you see snapshots of the organ at different pressures. And what I want you to emphasize here, the pressure that we are talking about are the pressure within pressure cooker, so 12 psi, 100 kPa. And so you can compare here the strain on the top, you have the strain along the circumference of the vagina, and in the bottom is the strain along the length of the vagina. And what you can see on the average is reported on the top is you have approximately 20% strain in the basically circumferential direction and half, 8% in the longitudinal direction. We can also measure changing curvature and so see how the organ change shapes during these deformations. And we can clearly see there is a difference between the change in the circumferential direction and in the axial direction, and it's reported with these two different type of lines, dashed versus continuous. And you can also see how the curvature doesn't change along the length of the vagina. So this is the, I apologize about this. About the basically along the sides of the organ versus the curvature around the circumference. And so here are some numbers just to get an idea. The maximum pressure that you can achieve within the organ is half the pressure that you have in your tire. So it's pretty high. At that pressure, the vagina is going to break. And usually the strain is around the 12% in the circumferential direction, the hoop direction, while it's half in the axial direction. And so you can see also that the stiffness of the organ is very different in the two different directions. So it's much steeper along the length and more compliant along the circumference, which, it makes sense given what the vagina has to accomplish. The other type of test that we do are tests where instead of just inflating the vagina and measuring the deformation, what we do is we apply a constant load, so we don't increase the load, and see if the vagina continues to deform. So these are called creep tests, and you can see some experimental data here. And what we do is tests where we take this organ and we inflate one time, and then we wait, right? And the vagina basically continue to increase creep, right? Continue to increase in diameter and length. And then we repeat the load. So imagine, and what we're trying to simulate here is what will happen if you have a baby moving through the vaginal canal, where you have the head of the baby, which represent the pressure, the load, right, that you're applying to the organ. And this load is basically increasing as the baby is moving through the vaginal canal. And so what we show in this data is that the first time the vagina is subjected to a load, it strains significantly. And then the second and third time, it continues to strain, but the strain level that you reach is lower. And you can see from this curve, first creep versus the second and third creep in both directions. This is the axial direction, and this is the circumferential direction. We also look at how fast basically the vagina can deform. So we call this the creep rate. And we can see that there is a significant difference, of course, between the two directions, and now it strains in the two directions. But also what we saw is that if you look at if you look at creep and you look at the primary creep rate, which is pretty much the slope of this region here, you have that the primary creep rate during the first creep is always higher than the second and third. But when you look at the secondary creep rate here, it's always higher during the second and third creep. So it basically deforms very fast at the beginning, and then the deformation is lower during the first loading, but things change during the second and third loading. All right, so we take all this experimental data, and then what we like is to basically develop basically constitutive equations. These are equations that basically relate the pressure we are applying experimentally to the strain that we are measuring. And the models are three-dimensional, really two-dimensional here, because we only have data that is two-dimensional. And so you can see here we account for M and N represent direction of fibers, which, by the way, we can also look at, and I'll show you a few images later on, but these models can actually capture creep in the two directions, so in the longitudinal circumferential direction, as well as first, second, and third creep. So we basically have a model that can capture all our experimental data. So we also looked at how, when you have what you're worried about many times when you have vaginal delivery is basically tearing. And so 80% of women experience some degree of tearing of the vagina. And so what we want to find out is why some women experience tearing and some do not, and which type of tear will heal by themselves versus which one actually will propagate to the point that cause a serious problem to the mothers, okay? And so to do this experiment, what we did is we use again mice, but this time we impose a little tear, which we know from previous experiment where it happens and how it happens. So we have, already tissue for which we know the tearing behavior. But what we want to see is how the tear propagates. So once you have a hole, you have a cut within the vaginal canal, how does that tear propagates? And so we use a similar experimental setup here. The only difference that we cannot just push water inside the organ because the water is going to leak out given the hole, right? So we basically have to create a kind of catheter. We make our own little tiny balloons that we use and insert in the vaginal canal to actually inflate the organ. And that's what you can see from this image here. And so things that we see are, we can measure pressure versus normalized tissue volume. We can also measure the axial load, so how much load is applied in the axial direction. But what I want you, from all this data, what I want you to look at here are two things. I have this TP initial, which represents the basically when I see that the tear, it starts to propagate. So if I have a hole within a tissue and I'm pulling the tissue, right? I'm stretching the tissue, clearly the hole is going to get bigger. That's normal. But that's not tear propagation. For me, tear propagation would mean that that hole is actually breaking. I have new surfaces that are being created. And clearly then things will start to, the tear becomes bigger. It's not just being stretched. And the other thing that is important is this maximum pressure, which is what you see experimentally, and happens with us as well, right? Is that even though you have tears, the vagina can continue to support load, sustain load. But if the load is too high, when you reach a maximum pressure, what you see is that the organ is immediately start to break. So you have that very, very fast, quick propagation of the tear. And so experimentally, we saw that at 40 kilopascal, we start to see that propagation of the tear, and at 50 kilopascal, the organ is completely done. So the tear, the tear starts to propagate so fast that it's almost impossible even to follow it. But if you look at the volume that the vagina can sustain at that point is 2.7, the original volume. So it means that you had initial volume and then it's almost 2.7 the original volume, and that's when you start to see the propagation of the tear. And in terms of this maximum pressure, you reach it when you have 3 times the volume, the original volume of the vagina. With strains that are very, very high in the up direction, you have 70%, 80%. So these are very, very high strain. In the axial direction is much smaller. All right, so we can then, with our technique, measure the strain all around the tear. And here I just want to say this, is that when you look at this hole and you look at this, these circles that I basically drew around the holes, what you see is when you go from that initial TP initial, that initial propagation to the maximum pressure, you have a lot of strain on the top and the bottom of the tear. So that the tear on the top and of the bottom is really stretching quite a bit. But if you look at the side of the tear, is the strain in the longitudinal direction that changes significantly when you go from that initial tear propagation to that maximum pressure. And so we can collect all sorts of data here. You can see, for example, here we measure this propagation of the tear, this delta AC plus delta AI versus the volume. And you can see very, very slow propagation. And then suddenly propagation becomes very fast. And you can see it with the pressure versus tear propagation. So you reach the maximum pressure and you have very, very small changes in the length of the tear, let's say in the tearing. And then once you reach that pressure, the propagation becomes very, very fast. Here's what you see when you look at the vagina. So you have a lot of collagen fibers, and this we can measure. We can look at them using second harmonic generation. So the fibers are very wavy, and you can pretty much look everywhere, the proximal, mid, and distal region. So those are the region closer to the cervix and away closer to the outside of the body. But once the specimens are loaded, you see that these fibers now straighten in the longitudinal direction. And then what we also see is that when you do have a tear, the fibers they're usually like along the tear, but at the tip of the tear, the fiber tend to reorient so that they prevent the propagation of the tear. What we do a lot of finite element simulations where we take the experimental data and we basically reproduce the geometry. We measure the mechanical properties, and then we start to reproduce the experimental data, and then look at different type of scenarios. What if the tail is smaller? What if the orientation of the tail is slightly off with respect to the axial load? So we can actually reproduce experimental data. And these are, for example, some example where we can reproduce the tail length versus pressure that we see experimentally. All right, so the other thing we need to keep in mind is the data that I show you now is data from tissue that is passive. So in other words, it has not been contracted, but we know that this organ is able to contract. And so we have done also experiments where we keep the specimen, we simulate the specimen in a high concentration of potassium to basically make sure that smooth muscle cell contracts. And we do what they're called isometric tests, where we take our specimen, we fix the length, and then after we fix the length within the bath, and here is actually the setup. So it's a biaxial, planar biaxial in this case. We keep in a bath, we fix the length, and then we contract. As we contract the organs, the tissue wants to basically contract, right? But it's fixed length. So therefore, what you see is overall the strain is 0 because you have the specimen is fixed, but you have locations where the strain becomes very high, positive strain, and other regions where you have very high negative strain, so that the average is 0. And so we can measure those differences. Here is an example of what you will see at a stretch, equibiaxial stretch of 1.1, where we just change the bath in which the specimen is immersed with KCl. We add KCl, and this is after 60 seconds of the simulations. And one thing that we found in this case is that the strain that is used just to the activation, so there is no strain here that I'm measuring due to the load. This is only strain due to the activation. And in this case, the strain in the axial direction is significantly higher. So basically the vagina, when you are applying a contraction, it wants to contract more in the axial direction than in the circumferential direction. So you have higher strain in the axial direction. And so this is just the data showing that the significant difference and actually the between forces and strain in the two directions. And we also do some immunohistochemistry, tissue clearing to basically look at why do we have stronger contraction in the axial direction. And what we saw is there is more smooth muscle oriented within the axial direction of the vagina. All right, so now some of you may ask, fine, you took this specimen outside the body, but reality is the vagina within the body is undergoing very, very different deformations, right? Because everything is now constrained. And so we wanted to find out what happens to the organs under normal conditions in the body, but now at this point, what we're looking at is what happens during pregnancy. And so we did MRI studies where we take mice and we basically follow them. This is a longitudinal study. We follow them throughout pregnancy. We start a non-pregnant, mid-pregnant, late pregnant, postpartum, and 3 weeks postpartum. And then we can have beautiful images like this. This is just example of some of the images that you get. This is a cross-sectional view of the vagina for the mouse 1, mouse 2, and mouse 3, and again at different time points. And so you can really perform all sorts of measurements. And by the way, just for the sake of it, so this is basically, I don't know if you can, this is the reproductive tract of a mouse. This is the vagina right here and the uterine horn. And this is the actual size. And this is what happens to the organ after pregnancy, right? So late pregnancy, you can see this is the vagina right here. And this is how big now it became. So very, very different, right? So a lot of remodeling of the organs. And so if we look at the vagina alone, we looked at everything, but the vagina alone, we can see that in late pregnancy, the length of the vagina increases as well as the volume. All right, so the next thing I want to talk about, I think I still have a few minutes, is how do we model all this information? So what we're trying to do in our lab is we want to have models that can actually be used in clinical setting. So which means you want models that are very fast. They have to run very fast. They have to be very simple so that, you know, anyone who's not an expert in finite element can actually use them. And they have to be you have to be able to run the model on a local computer because you don't want to send your data because there is always the possibility of, you know, people stealing your data and having data all over the world, right? So you want to keep everything local. And so what we have tried, this is just very, very simple example. What we have tried to do is said, okay, what do we want to do? We have this very high, and this is a very simple simulation that I'm showing here. It's just a cylinder under pressure. So really not much, not complicated at all. But we have these full order models, and we can generate full order model based on experimental data. And we have a lot of variation in experimental data, which we can also account in terms of computational models. And then the idea is we start to change different things. For example, the fiber orientation in different regions, the stiffness of the fibers, the size of the specimen, because it is different. And we have a bunch of simulation that we have, a lot of simulation. The question is, do we need all these simulations? Like this is an example of a matrix with all the simulation that we have. Well, the answer is no, you don't need all the simulations, but how do we find out which one we need? And so what we use is reduced order models. So where the idea is, if I want to say that this is the FOM is denotes the full order models. If I want to say that this is a bull, perhaps I can just look at this image here and say that's a bull, right? Few lines can tell me that what I have is a bull. So the same thing happens with all your massive sets of data. It's like I have a lot of data, but perhaps I don't need all of it. I just need a few lines that represent the entire data. And so we use this technique to reduce basically the data that we have. And so we use first what's called the kind of the SVDs. Many of you may be familiar with the SVD, but the idea is you find the dominant modes of your data, and then you just use, depending on how fast you want your simulation to be, you use some of this mode. For example, for this set of data, what you can see that after 70 modes, there is not much difference in the curve here, indicating that that's enough. You can just pick 70 modes to have a good representation of your data. And then the other thing that we do, and again, I'm showing this without equations to make it clear, but I'm happy to answer questions. The other thing that we do is we use what's called the data-driven variational multiscale reduced order models. And so the idea is, once you pick your modes, is it possible that you can capitalize, that you can use this mode more to describe things, features that maybe are represented by higher modes? And just to explain you this, is like, this is the lowest mode, right? You have 11 lines that represent the bull. What if I want a higher dimensional ROM, like this one? So basically, a model that has a little bit more details. You can see here now I have the musculature, I have more about the reproductive tract of the bull, right? So the tail is a little bit different. Well, the idea is I use these 11 modes, and try to use those to still describe for example, the musculature, right? And so this is basically the idea of this DDVMS ROM. So basically higher modes information is described by lower mode, but you need to apply an offline transformation, okay? And so that's pretty much what we have done with our data. And you can see the comparison between the GROM and the DDVSM ROM, that within some range, the DDVMS ROM does better than the GROM. So basically you can capture good information. Of course, that comes with some time, as I say, the offline time that increases, as well as some online time. And this is just the error presented between the GROM and this DDVMS ROM, and also this idealized ROM that is just like the best possible case that you could get. All right, what we're interested in is basically the speed up factor, how much faster our simulations are, right? So you start with basically the finite element simulation that represent the full order model. And then you start to see, okay, if I use this GROM, I get basically my simulation that are on the order of 10 to the 6 faster, so significantly faster, which is what we want, what we're trying to predict very quickly if a woman has to undergo a vaginal delivery versus C-section. So we want something that is very, very fast. All right, so some final conclusions. So basically what we have shown, what I showed you today, is that the vagina is an organ that is highly nonlinear and is anisotropic and undergoes large deformation. In the passive state, the organs tend to deform more in the circumferential direction. But when it's contracted, what you see is that the deformation is significantly higher in the longitudinal direction. We also saw that tears usually form along the longitudinal direction as expected. And they basically propagate very slowly until reaching a maximum pressure, after which the propagation is very fast. We also show that the length of the vagina and the volume increases in late pregnancy. And if you use reduced order models, such as the GROM or the DDVMS-ROM, you can get faster, still accurate simulations of vaginal deformations. So what do we do with all this data? Oops. Okay. So basically the idea is if we understand the mechanics and the microstructure of the vagina, we can develop better models. And once we have better models, we can create computational models that can actually be used to make prediction about, the tearing or about C-section or selection of, one mode of delivery versus another based on the tearing. We can also create some biomedical device that can be used to actually prevent vaginal delivery. For example, think about dilation devices like the one you can see here. We're working on that in our lab as well. We can create graft material that have the same material as the native tissue. And we can also give surgical guidelines in terms of, how, if you have a tear and the tear orient in this direction, should you stitch it up or do you leave it alone? Like what's going to be better for the patient. Finally, I want to say this is not my work, but it's my students' work. So I acknowledge really their contribution. Some collaborators, as well as the funding source, NSF. And with that, I think I'm done, and I'll be very happy to answer any questions. Thank you.
Well, let's thank Raffaella for this really wonderful talk. So I can see currently 2 people have posted questions, and let's see. We'll probably only have time for one if the answer is longer. So Ria Samantha asks, I was wondering how tear propagation changes with first-time mothers for different ages in mice.
Yeah, that's very interesting. I haven't done that work yet. I haven't done that study yet. This is primarily my funding comes from NSF. And so it's always primarily on method development, right? But I'm trying now to look at also getting NIH, where you can have different group of animals. But definitely it does change. Especially not only for the vagina, but especially for the uterus. As you know, if you have a C-section, you can have 2 C-section, and after 2, the doctor will tell you, oh, no, you can't have more because now you have a scar within the tissue, and that scar can reform. It's dangerous for the mother. So definitely, the more pregnancy you have, the more likely you have, if you have tearing, the more likely you're more likely to develop another tear later on.
So there's another
but
question.
I haven't done the study.
Okay.
I haven't good.
Yeah, we can take another question, I think. So Helan Ananta Krishnan asked, thank you for sharing research, very insightful. So could you talk more on how you model tear propagation in the simulation? And second question is, on the wall clock time for one simulation, how much is the time saving with ROM? I guess the second is the answer.
Yes. So 10 to the 6 is the basically how much faster, right? If you have like 1 minute for the finite element, well, let's say, so yeah, 10 to the 6 is the how much faster they get, right? So definitely it's a significantly less time. In terms of how I do the finite element, so basically what you will do, you ask finite element, you have some damage threshold within your model. And so you basically say, okay, well, the stiffness of the fibers changes once there is some damage. And then the stiffness, you basically have a parameter that you multiply the stiffness for, and that parameter basically changes from 0 to 1. And once you reach breakage, right? So what you do is you delete an element. And so with the element deletion, that's how you include, you have the tear propagating.
All right. Well, thanks. That's really wonderful.